Welcome and good morning, everyone. So today we continue our lesson with um, cleavage and gastrulation. Okay. Now before that, apparently this cleavage, right, starts from fertilization. I forgot to mention it is the embryonic development. Okay, it is the fertilization marks the starting of the embryonic development, also known as embryogenesis. Okay, so embryogenesis is the first 2 months of development of an embryo following fertilization. Okay, or after fertilization, the embryogenesis kicks in or starts. So this embryogenesis took around 2 months. Okay, so consists of fertilization, cleavage, gastrulation, and organogenesis. So for today's lesson, we will be discussing about cleavage and gastrulation. Okay. Embryonic development or embryogenesis is initiated by fertilization and cleavage. Okay, now before that, so this diagram here, it shows you the fertilization process, and then this is the outcome of the cleavage, which is the formation of a blastocyst. Okay, and then gastrulation, which is the formation of the 3 germ layers. Okay, then organogenesis, which is the formation of the rudiment organs. Okay, right now let's focus on cleavage, which occurs straight after or immediately after fertilization. Okay, so cleavage is a cell division without growth, where the zygote rapidly divides into many cells with no overall increase in size. Okay, 30 hours after fertilization occurs, cleavage will partitions the cytoplasm of the zygote or embryo into many smaller cells called blastomeres through a series of mitotic cell division. Okay, during early cleavage, the zygote will be in 2-cell stage, as you can see from here, and then it progresses into 4-cell stage. Then the embryo progress, or the zygote progress into 8-cell stage, right? And then to morula, okay, or also known as early blastula, which is a solid ball that has 32 uniform cells, and each of these cells here inside this morula, these cells are the blastomeres, right? In the 2-cell stage, we have 2 blastomeres. Okay, in the 4-cell stage, we have 4 blastomeres. 8-cell stage, we have 8 blastomeres. On morula, we have 32 uniform of blastomere cells. Okay, and normally morula is formed on the 3rd day. Okay, and then cleavage will end with the formation of a blastula, right? So this is a blastula. So this is the end product in the cleavage. Now, this blastula, also called blastocyst in mammals. Okay, so if you come across the word blastocyst or blastula, it is the same meaning. Okay, now this blastula, right, or blastocyst, is a hollow ball of cells with blastocele. This is a blastocele. It is a fluid-filled cavity. Ruang kosong ni tapi ada fluid di dalam, right? It's a hollow ball of cells with blastocele, and it has embryoblast. This is the embryoblast. This embryoblast, also called the inner cell mass. Okay. Now, this embryo, right, is called blastocyst, right? So initially we have the zygote after fertilization, and then the zygote, right, enters the 2-cell stage. During this period of time, that zygote is already also known as an embryo, right? So now the outcome of the cleavage is a blastocyst, where we have the embryo or the zygote develops into a blastocyst. Now, a different look on the blastocyst, right? So you can see this is the inner cell mass, right? Then this is the trophoblast, okay? And this is the blastocele, ruang kosong yang ada fluid, okay? So you can see again trophoblasts, how they look like, and then the inner cell mass and the blastocele. Okay, students, please watch out with the spelling of the blastocele. Alright, so a quick recap here. Okay, so oogenesis, after oogenesis, fertilization, right? The cleavage starts here, right? Where apparently we get the blastocyst. Okay, now this is the blastocyst, which normally form on the 6th day, right, after fertilization. So fertilization is on the 0th day. Okay, so after cleavage will be implantation. You can see here, and this implantation occurs in the wall of the uterine or uterus. Now, implantation is the embedding of an embryo at the blastocyst stage into the endometrium of the uterus. So you can simply be defined as embedding of a blastocyst into the wall of the uterus. Okay. So the blastocyst must reaches the uterus first for this implantation to occur. Okay, so normally implantation occurs on the 6th to the 9th day, right? Normally it's on the 7th day. Okay, initiated by trophoblasts, right? So these trophoblasts, which I explained earlier, will secrete enzyme that can break down the molecules of this endometrium. Okay, so now after the molecules of the endometrium already break down, okay, this will allow the blastocyst to be embedded into the endometrium of the uterus. Now, the presence of this blastocyst will produce human chorionic gonadotrophin hormone, right? Mind the spelling here, or HCG, that stimulates the corpus luteum to continue produce progesterone and estrogen to maintain the uterine lining. Okay, next, the trophoblast, okay, when it's already embedded into the wall of the endometrium, what happens? This trophoblast will form syncytial trophoblasts, right? Can you see this syncytial trophoblast? Right, so it is derived from the trophoblast. So this is a syncytial trophoblast, right, that will further penetrate into the endometrium, and then this trophoblast also form the cellular trophoblast. Can you see here? This is a cellular trophoblast that will absorb and carry nutrient to the inner cell mass. So apparently this cellular trophoblast will absorb nutrients from the wall of the endometrium, okay, to the inner cell mass. And then this trophoblast, right? This trophoblast here also form lacunae and primary villi. You can see here the lacuna, right, and this primary villi, which eventually will form the placental tissues. Amniotic cavity is formed within the inner cell mass. Okay, you can see here. This is the inner cell mass, and then from here, amniotic cavity will form. Okay, within this inner cell mass. So you get this is the inner. This is the amniotic cavity. Okay, and this amniotic cavity is surrounded by amnion. Okay, this is the amnion. Inside this amnion is the amniotic cavity. Inner cell mass, right, will develop into embryonic disc or a flat disc. Okay, and then this flat disc, okay, is the inner cell mass develops into a flat disc or embryonic disc between the amnion and blastocele. Okay, between the amnion and the blastocele. So this is the blastocele, right? Oh, sorry, here, yeah, this is the blastocele. This is the amnion. So between this amnion and this blastocele, okay, is the embryonic disc. So this is the embryonic disc, and this embryonic disc is consists of epiblast. Okay, you can see this epiblast here, which is the inner layer, right? Inner layer of the embryonic disc, and then the hypoblast, which is the outer layer of the embryonic disc. So inner layer, epi means in, hypo, luar, okay, hypoblast, which is the outer layer of the embryonic disc. Okay, so apparently this embryonic disc also known as flat disc or blastodisc. Okay, blastodisc. Right, so we have epiblast and hypoblast. Next, the trophoblast will continue to expand, okay, into the endometrium, right, and four new membranes, okay, or extra embryonic membrane will arise from the embryo. Okay, these four new membranes or extra embryonic membranes, they are chorion. Okay, this is the chorion. Initially it was here, right? You have the trophoblast here. So from here the trophoblast expand, okay, and eventually form these four new membranes. Okay, so chorion, and then we have amnion, okay, which is from the inner cell mass. The amnion is here, that's why we have the amniotic cavity. Okay, we have the amnion. Then we have yolk sac from the hypoblast. Okay, so this is the hypoblast. From this hypoblast we get this yolk sac. Okay, and then we have allantois, okay, which is the extension of the yolk sac. Okay, and this allantois is derived from the epiblast. So with the formation of these four new membranes, implantation is completed. Now gastrulation begins. Okay, gastrulation begins on day 13. So gastrulation is the reorganization of the blastocyst, all right, into gastrula that has three primary germ layers, and these three primary germ layers are the ectoderm, mesoderm, and endoderm. Okay, or you can simply define gastrula is a three-layer embryo that has four embryonic membranes. Now, some epiblast cells, okay, will remain as ectoderm on the surface. Now, okay, this is the yolk sac, right? Okay, then okay, now we have this diagram. Initially, we have this diagram, right? So if we turn this turn this 90 degree, we get something like this. Alright, pusingkan ini sikit, tampak macam ni. So this is the epiblast, right? this is the epiblast. Now this epiblast, okay, some of this epiblast that remains on the surface will become the ectoderm. Okay. Now there are other epiblast cells that will move inwards. You can see how this epiblast, this part of the epiblast cells that move inwards. Right, it moves inwards here as well as further inwards here. Okay, the epiblast that move inwards here, right, through this primitive streak, okay, will form the mesoderm. This part will form the mesoderm, and this part will form the endoderm. Right, and this primitive streak is a very special characteristics, all right, in embryo, which marks the gastrulation process. Okay, so we need to say we can tell that an embryo undergoes gastrulation by the formation of this primitive streak. Right, so you can see how these epiblasts fall inwards to form the mesoderm and endoderm. So by the end of the gastrulation, okay, at the end of the gastrulation, the primary germ layers in the embryo have been formed. So we need to say embryo has these 3 primary germ layers. Okay, this marks the end of the gastrulation. So it is the ectoderm, the blue ones here, and then this is the mesoderm. Meso means tengah, ecto luar. Okay, and this is the endoderm. Okay, dalam tengah luar. Extra embryonic mesoderm. Okay, this is the extra embryonic mesoderm, which was formed earlier, right? And the 4 distinct extra embryonic membranes. Remember the chorion, amnion, yolk sac, and allantois. Okay, they are all here. Okay. Now surround the embryo. So I mean to say at the end of gastrulation, the embryo, like what I mentioned earlier, all right, has these 3 germ layers: ectoderm, mesoderm, and endoderm. Okay, and then the 4 extra embryonic membranes, which is the allantois, yolk sac, chorion, and amnion. Okay, as well as the extra embryonic mesoderm. Okay, so this is the outcome of a gastrulation, or how a gastrula looks like. Now, cells of the invading trophoblast. Remember earlier during implantation, trophoblast invaded into the endometrium. Alright, so the cells of the invading trophoblast, the epiblasts, right? Some of the epiblasts that still remains here, and the adjacent endometrial tissues, right, all contribute to the formation of placenta. So I mean to say placenta are formed from the invading trophoblast, epiblast, and adjacent endometrial tissue, right? This placenta mediates, right, the exchange of nutrients, gases, and nitrogenous waste between the developing embryo and the mother. Now, allantois will form the umbilical cord, all right, and then the chorion, right, this chorion forms the chorionic villi that will induce the endometrial tissue to form the maternal placenta. Okay, now class, you need to know the function of each of the germ layer: ectoderm, endoderm, and mesoderm. Not function, what they will eventually become. Ectoderm will become the outer layer of skin, hair, lining of the nose and mouth, and the nervous system, right? The endoderm will become digestive tract, respiratory tract, liver, and pancreas, while the mesoderm will become the muscles and skeleton. Okay, so this is the primitive streak. which indicates gastrulation. Okay, so these are further into the outcome of the ectoderm, mesoderm, and endoderm. Right? Please take note on this. Okay, until then, see you in the next lesson, which is about organogenesis, which is the final part of the embryogenesis. Goodbye.